Mitosis is the highly regulated process of the division of the nucleus. One cell becomes two, each with the genetic equivalent of the parent cell. The end result of the mitosis process results in 46 chromosomes, one set of 23 from each parent. Before mitosis takes place, the process of interphase occurs. Interphase accounts for 90% of the mitosis cycle. During interphase, the parent cell grows and copies its chromosomes in preparation for cell division. There are three phases of interphase: G1, synthesis, and G2. During the G1 phase, the cell makes more ribosomes and proteins so that it can grow to its proper size. The purpose of the G1 phase is to grow the cell. The second phase is the synthesis phase. During this phase, chromosomes within the cell are copied for the preparation of mitosis. The third and final phase of interphase is the G2 phase. During this phase, the cell replicates its organelles in preparation for mitosis. The G2 phase is also the last checkpoint for the cell before it enters mitosis.
After interphase is the metonic phase, or the M phase. The M phase accounts for 10% of the cell cycle. It is divided up into four phases: prophase, metaphase, anaphase, and telophase. In prophase, the chromatid fibers become more tightly coiled and condensed into discrete chromosomes. The nuclei disappears. Then each duplicate chromosome appears as two identical sister chromatids joined together at their centromeres and all along their arms by cohesion. The metonic spindles begin to form, which is composed of the centrosomes and the microtubules that extend from them. Then the asterids are the radical arrays of shorter microtubules that extend from the chromosomes. Finally, the centrosomes move away from each other, apparently propelled by the lengthening microtubules between them.
Metaphase is the longest stage of mitosis, lasting about 20 minutes. As seen in the picture, the two sets of centrosomes are now at opposite poles of the cell. The chromosomes lie on the metaphase plate, an imaginary plane that has equal distance between the spindle's two poles. For each chromosome, the kinetochores, a protein structure on chromatids, where the spindle fiber is attached during cell division to pull sister chromatids apart, of the sister chromatids are attached to the kinetochore microtubules coming from opposite poles.
After metaphase is anaphase. Anaphase is the shortest stage in the M phase. It begins when the cohesion proteins are congealed, which allows the two sister chromatids of each pair to part suddenly. Each chromatid becomes a full-fledged chromosome. The two liberated daughter chromosomes begin moving towards the opposite ends of the cell. As their kinetochores region, the chromosomes move centromere first. The cell elongates as the non-kinetochore microtubules lengthen. By the end of anaphase, the two ends of the cell have equivalent and complete connections of chromosomes.
Telophase is the last stage of mitosis. During this stage, two daughter nuclei form in the cell. Nuclear envelopes arise from the fragments of the parent cell's nuclear envelope and other portions of the endomembrane system. The nucleoli reappear and the chromosomes become less dense. After this stage, mitosis is complete.
Cytokinesis is the division of the cytoplasm, usually underway by late telophase. So the two daughter cells reappear shortly after the end of mitosis. In animal cells, cytokinesis involves the formation of a cleavage furrow which pinches the cell in two.
The cell cycle is controlled by checkpoints. A checkpoint is a control point where stop and go ahead signals can regulate the cycle. These signals report whether or not crucial cellular processes that should have occurred by that point have in fact been completed correctly. The G1 checkpoint is the first of three checkpoints during the cell cycle. At this checkpoint, if a cell receives a go-ahead signal, the cell continues on the cell cycle. If a cell does not receive a go-ahead signal, This cell exits the cell cycle and goes to G null, a non-dividing state. The G2 checkpoint is the second checkpoint in the cell cycle that occurs right after the G2 phase. This checkpoint ensures that all of the chromosomes have been replicated and that the replicated DNA is not damaged for the cell to enter mitosis. The M checkpoint is the final checkpoint of the cell cycle that occurs in anaphase. This checkpoint determines whether all the sister chromatids are correctly attached to the spindle microtubules before the cell enters the irreversible anaphase stage.
The internal signals in each cell cycle checkpoint has fluctuation in chemicals. An example is mitosis promoting factor, also known as MPF. The external signals in each cell cycle checkpoint is when cells fail to divide if an essential nutrient is lacking in the cultural medium. Even if all other conditions are favorable, some will not divide unless a specific growth factor is present. An example is when fibroblasts have a type of human growth factor known as PDGF receptors on their plasma membranes. Binding of PDGF molecules to receptors triggers a signal transduction pathway that allows the cell to pass the G1 checkpoint and divide. When a cell goes through mitosis, there are various checkpoints where the cell makes sure it's healthy and ready to divide. If a cell passes the checkpoint, it is more likely to complete the cell cycle and divide, or it can undergo apoptosis, which is death. Apoptosis occurs when the cell senses that the DNA is damaged. When the cell senses the damage, the cell tries to fix it. But if the damage is too much for the cell will kill itself so that the mutation is transferred to daughter cells. If apoptosis does not work, cells with damaged DNA are able to divide, which leads to accumulation of further mutations causing cancer.